All right, class. Welcome back. We are studying the brain and the cranial nerves. Excuse me, I got some crud still working out. So last time we started looking at the anatomy of the brain and all the four principal parts here. The cerebrum in the purple, the cerebellum in blue, the diencephalon in brown, and the brainstem in green. Okay, we walked through the overall anatomy of those different various parts. All of these are the brain, okay? And they all have unique locations and functions that we go on to elaborate a little bit. So just to continue the review, okay, make sure that you know those parts of the brain, the lobes, the parts of the brainstem, etc. The areas of gray matter and how we compared from the spinal cord to the brain itself. And eventually we will get into some more specificity about the various regions around the brain. The first thing I said was that in general, if you think about sensory versus motor function, okay, sensory is the first part and motor is the second part of that U shape, okay? There's a anatomic organization to that. Most of the back of the brain is sensory and most of the front of the brain is motor and higher executive function, okay? Then I started to describe this concept of the triune brain, okay? And this is very foundational, fundamental understanding of how the brain works, okay? With respect to the types of things that the brain does, okay? The most core, vital, foundational things that the brain does is to keep the body alive, okay? Vital means pertaining to life. So vitality means how do I stay alive? Well, we need to control heart rate and breathing and metabolism and body temperature. All those homeostatic, gotta have them or we die things are in the brainstem, okay? If you injure your brainstem, you will die. You can have deficits and damage and strokes and other problems in the cerebrum, and you would have some cognitive deficit or a deficit in motor function or sensation, but and you could still live a reasonable life. You could stay alive, okay, outside of the ICU. But if you have a problem in the brainstem, that is life-threatening. Okay, so that's the first thing. Okay, the lizard brain or the reptilian brain is the most core foundation of staying alive. Okay, the mammalian brain or the limbic brain is more about social interaction, how we regulate mood and memory, emotions, memory, habits, and attachments. Okay, so social interactions, okay, and really kind of communication as well to some extent. And then lastly, the human brain, or the most advanced and higher developed part of the brain, which we call the cerebral cortex. And really the neocortex is this outermost layer, and really the front of the brain is the prefrontal cortex, which I said is kind of higher order functioning, reasoning capacity, personality, planning, problem solving, etc. Okay, so triune brain is really a key way of understanding the brain before we get into the nitty-gritty of the anatomy. All right, so the last thing we had left off with was we'd gone a little bit through some of the developmental terminology. I'm not going to ask you about it, but I'm kind of introduced you to the terminology. And then we talked about the ventricles. Excuse me. The ventricles are these hollow chambers filled with CSF, and they have a very unique shape, and they lie really inside of the structures of the brain, okay? All of these are hollow chambers, the right and the left lateral, the third ventricle, and the fourth ventricle is what we talked about. Here's a nice 3D model of that. All right, so we're gonna continue our study of the brain, looking at the ways that we protect the brain. What are the ways in which the brain is protected? We studied it in the spinal cord. We used the analogy of the car crashing into the tree, and we have the structure of the car, okay, like the roll cage, as it were. We have the seat belt, we have the airbag, we have all these parts of the car that protect the guy in the car, right? So in a very similar fashion, we have structures and fluids and connective tissues that protect the brain inside of the cranium, okay? So the bones of the cranium, the skull, we learned about all the skull bones, frontal, parietal, occipital, etc. Okay? We talked about the cranial meninges, which are the same layers as in the spinal cord, so it's going to be kind of easy to memorize. And in the CSF, we've also memorized as well. Talk about the fluid that lives in the subarachnoid space. Okay? We mentioned the blood-brain barrier when we talked about the glial cells. Remember the astrocytes. Astrocytes are the star-shaped glial cells that provide the blood-brain barrier. So we're going to talk about that a little bit. It's more of a biochemical protection to the brain tissue. The interface between the blood and the tissue itself is guarded, so stuff can't get from the blood into the brain tissue unless you go through these special cells in the barrier. All right, so here's a nice picture of the brain inside of the cranium, okay? And we have the meninges that we'll look at in a little bit closer. We have blood supply, okay? Here's the brain stem, okay? And as we pass through foramen magnum, which would be, this is the hole here, foramen magnum in the bottom of the occipital bone, okay? And then down below it would be the spinal cord. Okay, so let's zoom in here to the top. Okay, we have obviously this cranial bones provide some measure of protection. Okay, like a helmet protects a football player, so the skull provides some measure of bony protection. But we have the dura mater, which is this outermost layer, then the dura arachnoid, and then the pia. Pia would be this lightest and most intimate layer that's close to the brain tissue. Here's the arachnoid, and these arachnoid trabeculae, which extend or span the distance between pia and the arachnoid. That space is filled with cerebrospinal fluid. Okay, so that's kind of a watery cushion in which the brain is kind of suspended, as it were. Now, it's not floating exactly, but it is cushioned by that layer of fluid. Okay, and of course, the arachnoid is attached to the dura is attached to the bone. And in fact, part of the dura mater is continuous with the periosteum. Okay, remember what the periosteum was? It is the outer layer of bone on the inside of the cranial bones. There's a thin layer of connective tissue called the periosteum. That's in fact merges with and continuous with the outermost layer of the dura mater. Okay, so connective tissue. They hold the skull to the brain tissue. Okay. All right, so here are those three layers. We've already talked about those. Helps prevent cranial trauma. Okay, so when you get into a car accident or you have a concussion, okay, that means that the brain has been jarred. There's been an extreme change in momentum that causes the brain to kind of slam into the inside of the brain. Not very good. But these tissues are meant to prevent and to minimize that jostling of the brain tissue, because brain tissue is like gelatin. It's very delicate and soft. And so if you hit it hard, you can cause cellular damage. And cellular damage leads to cellular death, and then you have loss of function. So the fluid, the connective tissue, the bone, all of these things are meant to protect the brain. Okay, so here's a kind of a coronal section. You can see here's the right hemisphere and here's the left hemisphere. Here's the longitudinal fissure. Here's the bone, the scalp, the skin. Okay, and then two layers of periosteum. This inner periosteum is continuous with and actually becomes part of the dura mater. In purple is the arachnoid, and in blue is the pia mater. Okay, so you can see in these purple trabeculae, which are the arachnoid mater. We see these little outpouchings of arachnoid into this blue space, okay, through the dura. Those are called the arachnoid villi. We will talk about those in just a minute. It is a way of, it's kind of like a release valve for CSF to escape and to become venous return, okay, venous blood supply. So we will talk about how CSF returns to the blood here in the dural sinus, okay? But these are the different layers here between the brain and the skull and then the scalp above it. Now, there's some special dural folds. I'm not going to spend a lot of time here. I'll just briefly announce the terms: the falx cerebri, the tentorium cerebelli, and the falx cerebelli. Let me take you to a picture here. Okay, this is a infolding of the dura mater. You can see this structure in blue. Whenever I see the word falx cerebri, for some odd reason, I think of mohawk. Falx, mohawk. I don't know why, but it has this kind of shape like a mohawk is like this, right? And so that's the orientation. It's actually deep inside of the skull, but it's in this orientation. It's a sheet of connective tissue that separates the right and the left hemispheres, okay? And it extends into the longitudinal fissure, okay? That's called the falx cerebri. And then there's a flat one that runs in this kind of transverse section. It really is separating the cerebellum from the cerebrum above, okay? That's called the tentorium cerebelli. And then the falx cerebelli, again, is a very thin sheet, but again, it's still in this sagittal plane. Okay, so falx cerebelli versus falx cerebri. You can see the word cerebrum and the word cerebellum. Okay, so it's the vertical sheet of connective tissue that separates the right and left hemispheres of the cerebrum and the cerebellum. Okay, it's just this small little sheet right here. And then connected to those are these sinuses, okay? You can see these blue structures. Those are actually part of the blood supply, okay? And so we'll get to blood supply in a little bit, but in a kind of a very generic and simple way, you can think about blood supply in the brain as entering through the bottom in the middle, and the blood enters up into the brain from the middle, and then kind of percolates through the brain, and then ends up on the outside of the brain. Okay, so up through the middle and center, the blood percolates through the brain, and then once we've delivered oxygen and goodies, now we have turned the blood from red to blue. Okay, that's not really true, but that's how the diagrams are drawn. We have oxygenated blood and then deoxygenated blood, and then when the blood is here on the outside, kind of drains posteriorly and then out down through the jugular vein. Okay, so inside to outside and then back and then down through the jugular. Okay, that's the blood flow. We'll get to that later. Connective tissues are closely associated with those major vessels. Okay, so let's talk a little bit more about fluid flow in the brain. Okay, we already talked about CSF. This is special fluid. It's basically like fancy water. It's like plasma in the blood. Okay, but no red blood cells. Okay, we do not have any red blood cells here. But it has all the functions of blood. That is, it's distributing nutrients and gases and removing waste products and circulating around the nervous tissue, okay? It helps to cushion the delicate tissues, it supports the brain, and it transports and distributes those nutrients, okay? Those are the functions of the CSF, okay? CSF is made by the choroid plexus, okay? CSF is made by the choroid plexus. What's choroid plexus? Well, choroid plexus is a specialization of the ependymal cells. Okay, remember the glial cells, the ependymal cells line the hollow chambers of the central canal, as well as the ventricles. Okay, and inside each of those four ventricles, there's an area where the ependymal cells get specialized, and that's called the choroid plexus. It's a special interface between the blood and the CSF, where blood is converted into CSF. That's what the choroid plexus does, is it takes blood and it changes it into CSF, okay? Keeps the red blood cells out, because we don't have any red blood cells, but allows for the fluid to move into the CSF. Okay? And it's continually making CSF all day long, okay? These choroid plexus makes about 500 mils of CSF per day, okay? Which doesn't sound like a lot, but when you realize that the overall space that is contained with CSF is already quite small, this happens to be quite a large volume that is continually being made. Okay. All right, so the circulation pattern is, I'll just show it on the picture. Okay, so here is a look at this. Here's our ependymal cells in pink, okay? And so the ependymal cells line the hollow chamber. This would be CSF here, and here would be some nervous tissue. Here's a blood vessel. Here's maybe astrocyte. Okay, has these projections that are creating the blood-brain barrier here. And so the nervous tissue is interacting with blood supply. Okay, so that's one fluid that's important in the brain. It's just blood, regular old blood. Okay, and so there's a special interaction here of nutrients and gases, okay, and wastes and CO2. Okay, the exchange is happening in the nervous tissue that way. But additionally, we have CSF, and these same processes are happening in the CSF as well. Okay, there are unique differences between blood and CSF that make CSF necessary. Okay, it's too complicated to get into all of the reasons for that, but for now, know that blood and CSF are doing very similar things in the brain. Okay, so the exchange of CSF and the removal of waste is happening in these specialized cells here. This is the choroid plexus. Okay, an example. This is a drawing. Okay, so here's the ependymal cells, just plain old boring ependymal cells. And once we get into the choroid plexus, suddenly they change and they're more specialized. And over this membrane, we can now take blood flow that's going through this capillary, and we're going to change the plasma in the blood here, and we're going to make CSF. Okay, so the flow of fluid is always going to be this way. Okay, even though there is an exchange of waste. Okay, it's always a continuous flow of fluid in this direction. Okay, there is some absorption of waste products here, though. All right, so that's choroid plexus. You can see these little squiggly lines here. This would be the third ventricle. Here's the fourth ventricle. And the lateral ventricles, the first and second ventricles would be lateral here, but you can't see them on the sagittal section, mid-sagittal section. But this red squiggly line is representing the choroid plexus. So in each of these locations, you have specialized ependymal cells that are making CSF constantly, okay? So if we have constant supply, think about a spring, right? A spring is like where water's bubbling up out of the ground. So we're continually making CSF. Well, where does it go? Wouldn't it create pressure, and that would be a problem? Maybe. Okay, that's a good way of thinking about it. But the flow goes down the central canal through all these different aqueducts that connect the ventricles. All the ventricles are connected, and they connect to the central canal, which is here, runs all the way down the spinal cord. You can see there's a line of division suggesting that this is much longer. Okay, the fluid can leak out the bottom, okay, and surround the front and the back and all around the spinal cord. Actually, there's three openings in the brainstem just underneath the cerebellum. Okay, cerebellum is here. There's actually three openings, two lateral and one central opening, through which CSF is going from the fourth ventricle into this outer space that surrounds the brain as well. Okay, so positive production of CSF here. It's flowing through the ventricles continuously. It's flowing down the central canal. It's flowing around the spinal cord. It's flowing around the brain this way. You always have this cycling of CSF always being made. And the CSF ends up here kind of in this top region. And in this top region, around the outside of the brain, we have these arachnoid villi. Okay, remember these little outpouchings here? Okay, there's several of those little outpouchings in the top of the brain. Okay, it's called an arachnoid villus, and this is where CSF, okay, winds up here in the top and bleeds out or leaks out and actually becomes part of the blood supply again. Okay, this is colored blue because this is venous blood, not arterial blood. Okay, so this deoxygenated blood, okay, will then go back out of the head, flow down the jugular vein, enter into the superior vena cava, and then go back to the heart and become oxygenated again. Okay? So that's fluid flow. So we deliver blood through the internal carotid. Okay, the carotid arteries, we'll point those out a little bit later. Carotids are here, they're deep. Okay, that's where we get our carotid pulse. Okay, and then the blood leaves the brain through the jugular. Okay, so carotid up, jugular down. Okay, carotid up, fresh red oxygenated blood. Jugular is deoxygenated blue vein. All right. So blood-brain barrier, very important. We've already talked about that a little bit, regulating the appropriate exchange of small molecules and gases and other proteins. Okay, so it's a highly controlled barrier between nervous tissue and blood. Okay? Has specialized endothelial cells, it has those astrocyte podocytes, and you have the tight junctions, which are part of the blood-brain barrier. There are 4 special breaks, okay, where the blood-brain barrier has a special allowance or a special permeability is increased, and they're normally for movement of hormones, okay? So blood-brain barrier means we have a specific barrier that does not allow stuff to go through. Well, if you have to move hormones, which are in many cases large molecules or even sometimes proteins, we gotta get that big protein through the barrier. So we have to have kind of a hole or a perforation or a change in the BBB so that the hypothalamus and the pituitary and the pineal gland can secrete materials into in and out of the blood. Okay, so the blood-brain barrier has these unique locations where hormones and other small signaling molecules can pass in and out. Okay, so it can be kind of a liability, but again, these regions are, again, very highly regulated and controlled. Okay, so they're protected. Choroid plexus is another of these kind of exceptions to the blood-brain barrier because it's where the CSF is produced. All right. So, we talked about the parts of the brainstem and the medulla oblongata, and we're going to go through a little bit more detail. I'm just going to, again, reiterate on the structure here and just mention a couple structures as we go through another walk through some of the anatomy here, and just remind on structure-function relationships. Okay. Remember, I said brainstem is important for what? Staying alive. Okay, so brainstem has lots of functions for autonomic reflexes and visceral function. Okay, our visceral functions like keeping our heart beating, keeping our lungs full of air, okay, and regulating body temperature. Okay, there's very special nuclei in the medulla for regulating those activities. So here's a look at the whole brainstem from here to here. Here's the medulla, here's the pons, here's the midbrain. Okay, medulla oblongata, pons, midbrain. On top of the midbrain would be the thalamus, and then the hypothalamus would be here, epithylamus would be the back. Okay, so this is the region that we're talking about here in the reddish orangish color. Okay, below that would be spinal cord. So what does that mean right here is foramen magnum. Okay, foramen magnum. And you can see these little stalks that are kind of sticking out. Those are cranial nerves. Okay, and you have always a pair, a right and a left cranial nerve, 1 through 12. We'll walk through those later. But you'll see CN, or N here in this case, and you'll see a Roman numeral. Okay, the Roman numeral are going to be the cranial nerves. Okay, you can see all of them here. And generally from top to bottom, they're numbered. Okay, 1 through 12, although it is a little bit more complicated than that, but generally top to bottom, 1 through 12. All right, and so here's a ventral look at the brain. Okay, right now, if you were looking at me and I tipped my head back like this, and you're kind of looking up at kind of through my jaw and through my mouth up into the bottom of my brain, that's this view here. Okay, so here's the brain stem. Okay, this is the olfactory bulb that's going to be right over your nose. Okay, that's for smelling. Okay, you have a number of these cranial nerve stubs or stumps or branches here that would then extend out, and these red and blue lines represent your cranial nerves. Okay, 1, 2, 3, 4, all the way down through 12. Okay, we'll come back to that in just a minute. I'll just introduce right now. I wish I had a marker. I didn't bring my marker today. Oh, it's not great. It's not great, but I want to introduce the mnemonic because I've kind of whipped through it a couple times, but I've never really said it slowly. Just write it down and say it once, and we'll come back to it on our last lecture. Okay? So let's just make a note, a list here, 1 through 12. Oh, it's not going to work. My marker is not good. Marker is not good. I don't like it. change of plans, we'll do that later. Okay, the mnemonic is O, to touch and feel, very good velvet, heaven. Okay, O, to touch and feel, very good velvet, heaven. Okay, I'm going to go through that mnemonic later. It gives you the names of each of the cranial nerves. All right, let's skip over that for now. So here's an anterior view of the brainstem. Okay, medulla, pons, midbrain. And starting here, these red blotches here are meant to represent the olfactory bulb. That's cranial nerve 1. Okay, so if you go 1, and we go down here to this little stub here, it's actually going to come out this way. That would be cranial nerve 2, 3, 4, 5, 6, 7, 8, okay, 9, 10, 11, and then we wrap up here to 12, okay? So again, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So I've kind of traced the path 1 through 12. That's kind of the path that the numbering is, okay? Most of the nerves emerge at the boundary between midbrain and pons. You can see 3 and 4. 4 is actually, strangely enough, on the back side. It's the only cranial nerve that's on the back side of the brainstem. Okay, so you can see kind of peeking out here and here. It's because it's actually emerging off the back of the brainstem, which is super weird, but 4 is just a weirdo. Okay, so 3 and 4, and then 5 comes here right off the middle of the pons. 5 is actually a big one. It doesn't look like it right here, but this is the diameter of this nerve is quite large, called the trigeminal. Okay, 6, 7, 8, and 9 really are kind of in the boundary here between pons and the medulla. 9, 10, 11 are in the medulla, and 12 is several nerve roots in the medulla and actually coming from actually 12 is in the medulla. 11 actually has nerve roots that extend down into the spinal cord. Again, super weird, but the nerve itself emerges superior to foramen magnum, so we count it as a cranial nerve. I'm not going to ask you to know the locations of these cranial nerves, but I've just given you a general outline of how they are arranged as they emerge from the spinal cord for future reference. This is a posterior view. Okay, so this is, again, medulla, pons, midbrain, and then this is the back of the thalamus. This would be the pineal gland here. There's 2 bumps here called the corpora quadrigemina. Okay, it's on the 2 bumps, 2 sets of 4, 2 sets of 2, I should say, collect a total of 4, that are called the superior colliculi and the inferior colliculi. Okay, it's kind of a weird word, but those are part of the midbrain. And then we have this opening here, which is the opening. If we imagine that the cerebellum is sitting right here, okay, and we've removed that, we've sliced it off and we've removed the cerebellum, and now we're looking actually into the 4th ventricle, because the pons and the cerebellum, in between them is the 4th ventricle. Okay, so if we slice through here, we're actually looking through into the 4th ventricle. This red here is the choroid plexus. Okay, we have choroid plexus here of the 3rd and 4th ventricles. And let's keep going. Here's what that would look like on a real cadaver. Okay, these are your corpora quadrigemina, pineal gland, the opening to the 4th ventricle, cut surfaces here, your peduncles. We'll talk about those later. All right, medulla oblongata, 3 groups of nuclei. Autonomic nuclei, okay, for controlling autonomic function. You have sensory and motor nuclei of the cranial nerves, which I just talked about. The cranial nerves are emerging from the medulla, so you have nuclei where those nerves are, the sensory and the motor nerves are coming in and out of those spaces. And then it really serves as a relay station for sensory and motor pathways. That is, you have lots of white matter tracts that are just running through the the medulla, because they're relaying messages up from the spinal cord. An important structure that runs from the medulla into the pons and into the midbrain is called the reticular formation. Okay, that's shown in blue, the reticular formation. It's really important. Okay, it's a gray matter area embedded with embedded nuclei, regulates autonomic function, and it's important for maintaining consciousness. Okay, so we are conscious because the reticular formation is receiving the right types of stimuli. Okay, if we lose consciousness, okay, we go to sleep or we faint or whatever, reticular formation changes status. Okay, reticular formation, okay, autonomic function and consciousness is in the brainstem. Now, as I said, the medulla is a collection of nuclei. Okay, a nucleus is a collection of cell bodies. So, looks like a bunch of like pieces of candy that are kind of jammed in here, a bunch of like jawbreakers and like taffy that's been stretched out into these weird shapes. Okay, kind of these long oval shapes and really long finger-like structures. Just looks like a bunch of candy to me, kind of jammed together. But those are all individual nuclei. Okay, I have pairs on the right and on the left. We're looking here at the medulla. Okay. Now, if you remember, we were naming the white matter tracts. We said there was like the olivospinal tract. Okay, we talked about the olives, okay, or the rubrospinal. So this green structure is called the olivary nucleus. Okay, the olivary nucleus. We also have the nucleus cuneatus, nucleus gracilis, okay, the solitary nucleus. In red, we have the cardiovascular center. And then the yellow structure here, and the previous structure was blue, but in this diagram it's yellow, is the reticular formation. Okay, this long finger-like projection. What are all these structures? They're just collections of nuclei, okay? Collections of neuron cell bodies. So neurons are coming into there, synapsing with another neuron, and then the message continues, okay? So a nucleus is a collection of cell bodies, and that's where synapsing occurs. Ask you to know the locations, but when you hear the word olive, okay, it's kind of round, and anatomists named it the olives because it looked round, okay, strange, right? But if I twist to the side here, okay, here's a side view, here's the olivary nucleus, here's the cardiovascular center, here's the respiratory rhythmicity center, okay, the nuclei gracilis and cuneatus, and the yellow is the reticular formation. All of these are important nuclei that are inside of the medulla oblongata. Okay. Really important to emphasize here, cardiovascular center in red and the respiratory rhythmicity center in purple. Okay. What is that involved in? Keeping the heart beating and keeping the respiratory rate. Okay. How should the body regulate how fast the heart beats? And how much do the lungs inflate and deflate, okay? So there is a synchronicity to that. There's a way that if I exercise, both heart rate and respiratory rate go up, right? They go up together. It's not like heart rate goes up and we just keep breathing the same way. No, okay? So they go up together and they come down together, and they're correlated, right? These centers help manage what is the demand for oxygen. Okay, how's my head, how's my brain using oxygen? How are my muscles using oxygen? And can I sense the amount of CO2 and pH change and oxygen in the blood, and then keep telling the heart to speed up or slow down to make sure that I'm maintaining the proper oxygen saturation. Okay, because I need to get more oxygen in because I need to deliver it to my muscles or deliver it to my brain or to whatever. Okay, these nuclei are responding to the sensing those levels of oxygen and then giving commands to the heart and to the lungs, to the diaphragm, to maintain homeostasis. Okay. So these reflex centers, okay, cardiovascular center, control the heart rate. Respiratory rhythmicity center, control the diaphragm, which controls breathing. Okay, keep the blood moving. Okay, got to move the blood. and we got to get oxygen into the blood, as well as kind of glucose and fuel. Okay, so both of those things have to be correlated and is done in the medulla. All right, some other important nuclei, don't have time to spend on those. Okay, moving up from the medulla up into the pons. Okay, the pons also contains important respiratory rhythmicity centers. Okay, some of those are in the medulla, some of them are in the pons, some of them actually overlap between the pons and the medulla. Okay, because there's actually several nuclei that kind of span the boundary between the medulla and the pons. Okay, so important respiration centers are there, okay, as well as nuclei for different cranial nerves. So here's those apnoeistic and pneumotaxic centers here shown in purple. Those are for controlling breathing. Okay, so those are here. We have some of the cardiovascular centers are here, and the control of breathing rate is up in the pons. It's another nucleus. All right. So while we're in the pons, we're going to go backwards into the cerebellum. Okay, medulla, pons, go back into the cerebellum. Cerebellum is like a mini brain. Okay, has these. The vermis is kind of the central bump. It looks like a worm. That's what vermis means. Vermis. Okay. And the folia are these little tiny little bumps. It looks like the sulci and the gyri in the cerebrum, okay, but they're teeny tiny. They kind of look at, if you look at a gross anatomy of the cerebellum, it looks like ground beef. Okay, if you see ground beef just coming out of the grinder and have these little threads of meat, okay, it kind of looks like that. Okay, cerebellum. Those little variations are called folia. Okay, here's the ground beef. Doesn't it look like ground turkey? Maybe look more like ground turkey. Okay, but that is in fact the cerebellum. Okay, cerebellum sits at the base of the back of the brain. Okay, and is attached at the level of the pons. Okay, it's attached to the pons. The function of the cerebellum is. Hold on a second. Where am I going? Adjusts and coordinates postural muscles. Okay, so I said fine-tuning motor function. Okay, fine-tuning muscle movement. So coordination of like throwing a ball or walking or doing any sequence of events muscularly. Okay, and getting them to work in a nice smooth fashion. That's what the cerebellum does. Okay, fine-tunes motor movement and also motor reflexes. Adjusts and coordinates postural muscles, fine-tunes conscious and subconscious movements. Okay, that's the anatomy. Two important things you need to know here. Purkinje cells are special cells that are found in the cerebellum. Okay, they look like this. Okay, take a look. This is kind of a special fan-like shape. These are very unique cells. They're not found other places in the body. They're found in the cerebellum. They're very wide and flat. Okay, so this is a face-on view, and this is a side view. So they're a very unique shape. They have a round cell body, and here's the axon here. Okay, so you have all these dendrites. Okay, they feed into one body, and then there's an axon. So here's the way that they would be oriented. Here's the body, here's the dendrites, and the axon would be coming this way. Those are found inside of the cerebellum. Okay, those are called Purkinje cells. Purkinje was a dude that did a lot of anatomy stuff. So you're going to see this name in other places in the body, okay, in the heart. But Purkinje cells are special fan-shaped neurons in the cerebellum. Okay, you should know that term, and that's where they're located. Okay, the arbor vitae. Arbor is tree. Vitae means life. So tree of life. This kind of branch-like appearance is what gives rise to the name. Arbor vitae means tree of life. It's the appearance of the white and the gray matter in the middle of the cerebellum. All right. If you have disorders in the cerebellum, if the cerebellum is not working correctly, you can have lots of types of disorders, but one of the common ones is called ataxia. Ataxia is a dysfunction of movement. Okay, dysfunction of movement, and that kind of makes sense because if what the cerebellum is doing is fine-tuning and coordinating motor function, that is how our muscles enact movement, okay? If it's not working right, then I'm gonna have a tremor or a disability in moving my body, okay? There's lots of ways that would manifest, okay? Often walking, okay? So ataxia is, taxis means to walk or to move forward, okay? So we think about taxia as like a taxi from an airport is to move you from the airport to go into the city or to go home, right? So ataxia, the A there means no taxia, like failure to do taxis. The taxis means moving, walking, and doing it smoothly. Ataxia means like failure to limp or to jitter or to have a tremor or to have a lack of proper movement, okay? So that can take place in the speech formation, in hand and eye movement, in limb movement. There's a lot of ways in which ataxia can present, but it can result in dysfunction from the cerebellum. Alrighty. So we were here in the medulla, we went up into the pons, we went back to the cerebellum, now we're back up and we're moving up into the midbrain, okay? Midbrain. Midbrain has the tectum and the tegmentum, okay? So here's a sagittal section, okay? Sagittal section, just like this sheet here. We're zooming in here on the midbrain, which is this section right here. Here's the pons. The tectum means roof, okay? So like a roof above you. In this case, this means roof, so it's the back and top of the cerebral aqueduct. Here's the fourth ventricle, here's the third ventricle, and here's the cerebral aqueduct. And so the tectum is really, there's actually two little bumps on here that are part of the corpora quadrigemina, okay? The superior colliculi and the inferior colliculi. In the light blue is the tegmentum, okay? Tegmentum includes the red nucleus and the substantia nigra. I will show you, if you take a section right through here, it's a very famous look through the midbrain, and I'll show you, this is the posterior view. So here's the red nucleus, here's the substantia nigra, here are 4 bumps, which are our corpora quadrigemina, here's the pineal gland, okay, and here's our good friend, the reticular formation. Keeps changing colors on us. First it was blue, now it's yellow, and now it's orange, but reticular formation is there. Okay, so here's that section right through the midbrain. Here's the cerebral aqueduct, here's the tectum, okay, superior colliculus, these little bumps. And then this is the red nucleus, okay? Remember the rubrospinal tract? Okay, we talked about the white matter tract. Rubro means red. This is the red nucleus. It's just red because there's lots of capillaries there, but it's a special area in the midbrain where there's a special collection of nuclei, okay? Substantia nigra means important black stuff. Literally, that's what they called it. Important black stuff, substantia nigra, okay? There's a ring or a kind of a half circle of heavily pigmented nervous tissue, which they named substantia nigra. Important black stuff. All right. So this is what that would look like in a cadaver. All the color is gone because it has been fixed and chemically modified. You can still see a little bit of that pigmentation. There's actually melanin in there. Okay, substantia nigra. This is where would be where the red nucleus is. It's not really red anymore because again, it's fixed. Okay, here's the cerebral aqueduct. Here are the cerebral peduncles. Here are the 4 bumps that are on the back of the brainstem. Okay, so here's the section going through. You can see the 1, 2, 3, 4, 1, 2, 3, 4 of these colliculi. All right, so we've gotten through the brainstem and the cerebellum. Okay, now we're going to look at the diencephalon, the thalamus, and the hypothalamus. Okay, so in this semi-transparent view, we can see kind of through a portion of the temporal lobe here. We can picture the thalamus, which is sitting at the top of the brainstem. Okay, thalamus is a very important sensory relay station. Diencephalon. Okay, diencephalon includes all the words that have thalamus. Thalamus, hypothalamus, epithalamus. But we remember that epithalamus isn't used a lot, and we call it the pineal gland. Okay, so we're here in the diencephalon. The pineal gland is at the very back. Okay, it secretes melatonin. Melatonin is our sleepy time hormone. Okay, At nighttime, we secrete melatonin and it percolates through the brain, and the brain goes, oh, it's time to go to sleep. And the brain then can do important stuff that it can't do when it's awake. Okay, thalamus. All ascending sensory data comes through the thalamus. That's the one thing you have to know about the thalamus, is it's a sensory processing center. Okay, the thalamus is a sensory processing center. All the data that comes up hits the thalamus, synapses with other neurons, and then those neurons project out into the cortex. Here's the left thalamus, here's the right thalamus. There's a little gap between it, which becomes the third ventricle, okay? So the thalami are like two hard-boiled eggs that kind of sit next to each other, okay? And they just touch a little bit in the middle. It's called the interthalamic adhesion. So the thalami. Okay, the thalami, again, are a collection of cell bodies. There's a bunch of different nuclei here, okay? They're color-coded, the blue, the red, the green, the pink, etc. Each of these nuclei is a discrete collection of cell bodies that comes from different parts of the body. Some of these some of these neurons are coming from the eye, some of these are coming from the touch receptors of your hand, some of them are coming from sense of taste, some of them are coming from your sense of hearing, but each of them has sensory neurons that are coming to the thalamus, synapsing with another neuron, and then continuing that message and projecting up into the cerebrum. So here's a nice picture of that. You have sensory data coming up, hitting the thalamus, synapsing, and then projecting up into the different parts of the brain. And this is an important place to distinguish how sensory information enters the brain versus motor information leaving the brain. Okay, let's watch this. Okay, here's sensory. This is what we were just talking about. We have all these various types of sense receptors, right? It could be like a Pacinian corpuscle, or a Ruffini's, or a Meissner's corpuscle, or any just bare nerve endings like a pain receptor or temperature receptors. All that sensation is going to come into the spinal cord and ascend up the spinal cord. Is going to come first to the thalamus, okay, shown in green. Okay, here's the right and the left thalamus. Can project up into the thalamus. Synapse here. This is neuron number 1 from here into here. Neuron number 2 from here to here. And neuron number 3 up, projecting from the thalamus up into the appropriate part of the cortex. Okay, but the important thing is it goes through the thalamus. Sensory goes through the thalamus. Motor does not. Okay, notice here. So motor is descending signal, okay? Motor signal, we're having a thought, I need to move my big toe. I'm gonna tell my big toe to wiggle. So we're gonna have that thought up here, and this part of your motor cortex is gonna send that signal out. It never passes through the thalamus, okay? It goes right by it through this little tract called the internal capsule, okay? All motor signal bypasses the thalamus and goes down, and then we have this upper motor neuron in red, okay? All the different types of motor neurons would be kind of collectively shown. And then here in this bluish green color, you can see the lower motor neuron, just the beginning of it. It would be sent out to the hand or the leg or the whatever, okay? But I'm just pointing out at this point the role of the thalamus. Sensory, yes. Motor, no. All right, just to bend over the nail, this is another figure. It's in the same scheme as the other figure, but I just decided to show it as well. Okay, this would be sensory. Okay, it's lower limb sensory. Here's neuron number 1, neuron number 2, and neuron number 3. Notice that there's synapsing that occurs here in the thalamus, okay, and then projects up into the cortex. We call these first order neurons, second order neurons, and third order. So the first order is the one that's attached to the sense receptor. The second order is typically connecting the first order to the third order, and the third order is the one that winds up in the cortex, okay? So if you know the first and you know the third, the middle one is connecting somehow. All right, these are the various groups of thalamic nuclei don't have to know any of those, okay? But just know the thalamus has several different chunks, different regions that are coordinating sensory reception from different parts of the body. Okay. There's the thalamus in orange, okay, right here. Now, let's talk a little bit about the hypothalamus, okay? Sorry. Here's the thalamus, okay? Now we're gonna come into this area right here in green. This is the hypothalamus, okay? It reminds me kind of like a bird beak. If this is the head of the bird, okay, there's an eye here, I don't know, okay? Here's the bird beak. The bird beak is the hypothalamus, okay? Let's zoom in there. So, brainstem, midbrain. Thalamus would be here, this is mid-sagittal, so you don't see it because it's like I said, it's two hard-boiled eggs sitting next to each other. So in a mid-sagittal, you don't really see much of the thalamus. If you were just off-center a little bit, you would see the thalamus. But this is where the thalami would be. This is actually third ventricle. Right here is the bird beak right here, okay? So that's the hypothalamus. So here's a nice look here. Here's the thalamus. We've zoomed in a little bit. Here's midbrain, here's pons, okay? Here's the bird beak. And again, it looks like, to me, if this particular figure looks like you took a bunch of like starbursts and you kind of like jammed them together and made this kind of candy, taffy mush ball, okay? They're collections of nuclei. Again, there's collections of neurons coming in here, synapsing, and then that signal is going to a very specific location, okay? This is the hypothalamus. What's the hypothalamus doing? Okay. Here. Hypothalamus provides subconscious control to skeletal muscle, controls autonomic functions, it coordinates the nervous and endocrine system. That's really important, okay? It's the interface between how the brain is working and how the endocrine system is working. And it's really a way of initiating endocrine control. Okay. Endocrine control is the control via glands and hormones. So the hypothalamus takes brain signal and starts telling it and forming it into glandular hormonal signals, okay? Hypothalamus. Also does important things like it secretes its own hormones. It has basic homeostatic functions like controlling urges like hunger and sex and mood and like happiness. Okay, your sense of well-being and happiness actually comes from the hypothalamus. Okay, body temperature, very important, is controlled in the hypothalamus. All right, limbic system. Okay, limbic system. If you remember the triune brain, we have the core lizard brain, we have the mammalian or the limbic brain, and the neocortex. Okay, the limbic is this middle one. Okay, it's kind of what's referred to as the emotional brain. Okay, the emotional brain, emotion and memory are what you need to know about the limbic system. Okay, emotion and memory. So often we form memories when we have an emotional experience, right? You think back to the most memorable times in your life. They've been really, really scary, or really, really happy, or really, really angry. The emotion encodes the memory, right? Because you don't think like just the blah parts of your life are not memorable, okay? Like just driving to school every day, or whatever. Like you just don't remember those. You remember the things that are encapsulated with an emotion, okay? Whether it's joy or anger or frustration or fear or whatever, okay? So it makes sense that memory and emotion are encoded together. Right? The amygdaloid body is the center for negative emotion. Okay, so frustration, anger, fear are what the amygdala does. Okay, so the amygdala is right here. It's at the tip here. The amygdala is at the tip of the temporal lobe here. This is the medial part of the temporal lobe. Temporal lobe would be kind of coming down like this. Okay, this would be the lateral fissure. Frontal lobe, parietal, occipital, temporal. And this is all deep, okay? These are all deep structures. So one important thing that you should know about the limbic system is that it is a loose network of structures that have a common function, okay? And that is for emotional processing and memory, okay? It does a lot more than that, but that's what I want you to know, okay? So these structures here, the cingulate gyrus and the mammillary bodies and the hippocampus, this is the hippocampus right here, okay, in red. The amygdala is here, it's kind of an almond-shaped nodular structure at the tip, okay, and the hippocampus is here. Hippocampus, super important for memory formation. If you take away somebody's hippocampus, or you have a stroke, or you have a tumor or something in the hippocampus, you cannot form new memories, okay? So hippocampus, important for memory formation, super important, okay? All right. Going into the cerebrum a little bit, okay? Going into the cerebrum a little bit. I'm running out of time, but I want to make a few more points before we close out for this lecture. Okay, cerebrum is the largest part of the brain, controls all conscious thought and intellectual function, processes somatic, sensory, and motor information. Okay, so remember, somatic means voluntary and conscious. Okay, this is kind of the paradigm I laid down for somatic versus autonomic. Okay, somatic means voluntary, meaning I tell my body what to do, and I'm aware of it. Okay, autonomic is the reverse, involuntary and subconscious. Okay, that's autonomic. So cerebrum is mostly somatic function. It's composed of various areas of white and gray matter. Okay, you can see the pattern is gray, white, gray. If we're going from outer to inner, we go gray, white, gray. Okay, so this outermost layer of the cortex of the cerebrum is gray matter. This inner layer here is white matter. And we get to the innermost part here, this is, again, gray matter. It's a slightly darker, I don't know why, but that is the thalamus, okay? Thalamus here is part of the gray matter, okay? It's where the nuclei are. White matter means myelination, and then gray matter, no myelination. Just a couple shots here. This would be the longitudinal fissure. Okay, this red surface here is part of the insula. I'll point out the insula on another slide, but it's one of the lobes that we say is a buried lobe. Look right here. Here's the surface of the cerebrum. So the parietal lobe and the temporal lobe. You have to dig into this little cleft right here to get to the insula. The insula is a buried lobe. All right. So a couple of spots here and then we'll be done. Okay, so this is a left lateral view of the human brain, okay? Left lateral view, here's the brainstem, cerebellum, and there's a couple important landmarks that you should be aware of, okay? When you look at the brain, you should always be looking first for these landmarks, okay? The easy one here would be this edge here of the temporal lobe, okay? So that's gonna show up in a little bit. Really one of the easiest ones from the front would be the longitudinal fissure, okay? So longitudinal fissure, you can't see it unless you look down or from the front of the brain, but it separates into the right and the left hemispheres. Then we have the lateral sulcus here, or the lateral fissure. Fissure or sulcus is fine, it's the lateral one because it's on the sides of the head. Separates the temporal lobe from the frontal and parietal, okay? This lateral fissure, sulcus. Then we have the central sulcus, okay? This is the central sulcus, okay? It separates frontal lobe from parietal lobe, okay? Central sulcus, key landmark, okay? It's not always easy to find, well, is it this one or is it this one? Honestly, I struggle to know exactly which one it is. It doesn't really matter. You just have to know that it's always in this position on every brain, okay? This one doesn't have a name, but there is a general boundary that helps us to say this is the occipital lobe, and that separates from the parietal and the temporal, okay? It is an amorphous boundary. There's no specific sulcus. There's nothing here that tells you very clearly where the occipital lobe is. but it's understood that the occipital lobe is most posterior, and then we slowly fade into the other lobes. Okay, so here are those regions as well. We will cover these in the last lecture next time. See you in class.